rhythm in dissociation from Carl Dijkstra's group. Like this is so it's been behavioral states, or like, well, just internal states in general, like cognitive states, emotional states, and behavioral states. They've been an increasing topic of interest in recent years as like more and more advanced technology to record large populations of neurons has become available. Because it's still like not very well understood, like, even things like fear conditioning has been studied for decades, but the state of being in fear, like how the brain generates that and what in the brain that corresponds to, is not really well understood. And that's basically true for pretty much all I kind of want to use a word other than state, but yeah, pretty much all kind of all emotions, or even things like hunger, we're beginning to understand a bit better. Like simple things like hunger and thirst, we're beginning to understand a bit better than more complicated states. But still, it's like these kinds of behavioral, cognitive, emotional states are not very well understood because we haven't had powerful enough tools to study them. And so this particular paper is really a tour de force because it finds a neural correlate of the dissociated state that it actually can also causally induce this state. And so I think this paper is going to have very far-reaching implications in neuroscience. And so I will, So I didn't actually know how to introduce this paper, so I just went to Wikipedia just to find what is the dissociative state. If any of you have done ketamine or PCP, then you'll know already what it is. But for those of you who do not have direct experience, it's used in psychiatry and psychology to describe a lot of experiences, different types of experiences, but what they all have in common is some kind of detachment from your environment or surroundings. It could be emotional or physical or some combination. Basically you're detached from what they call, what Wikipedia calls reality, and some of us might like to call consensus reality. And it's different from psychosis in this way because there's not actually a break from reality. It's just that the individual is dissociated. And so a lot of the times patients who have non-drug induced dissociative symptoms, like, describe it as a feeling of being outside of themselves, or watching themselves in the third person. This paper has a lot of different techniques. So I didn't put like an intro to everyone in the presentation, but please feel free to interrupt with questions, or if you want me to explain something in more detail, or to clarify anything. Okay. Oh yeah, also, and they have a ton of supplementary data, which I also didn't put in the presentation just because there's too much of it. But if you wonder if they did an experiment, please ask, because there's a good chance that it's in the supplementary data. Okay, so I'll get right into it. So they were doing this brain-wide imaging of activity in the mouse brain. And so on the top row of pictures is the experimental setup. So basically they have a head fixed mouse, and they kind of train the mice to let you fix their head under a microscope so that you can then image with the microscope. And then what they do also is they open up the scalp and they thin the skull. So what you see in B is like the experimental preparation where the animal's head is open and the skull is thinned. Or actually, maybe they just put something on the skull. Maybe they just put a chemical on the skull to make it transparent. I don't remember exactly what they did in this paper. And then in the second panel of figure B is a fluorescence picture of, and so these are transgenic mice, and they have sparse expression of a calcium indicator protein called GCAMP6 in their neurons. And so whenever the neurons are active, they'll light up. And so basically with this mouse with expression of this calcium sensor sparsely throughout the brain, you can kind of get an image of brain-wide activity. And so in C, they're just showing the kind of regions of the mouse atlas. So C is a schematic, a simplified schematic of the mouse brain atlas from the top of, like just as in the other views you've seen. And so I guess I should say this is the nose of the mouse. This is the back of the head and the neck, and just to give you some bearings. And so RSP is this area we're going to be interested in. That's retrosplenial cortex. And they also did other recordings from the visual cortex, which is here, the somatosensory cortex, which mediates your sense of touch, and then some more frontal regions, which are involved in motor and cognitive function. And then the posterior parietal cortex, which is an associative area of cortex, which means it's thought to be involved in associative learning. And so they'll compare the activity of the retrosplenial region to these other regions of cortex later in the paper to show that it's special. Okay, so basically they have this setup for whole top of where they can image activity in the entire surface, dorsal surface of the brain. And I'm not really sure. I think they might be like doing a screen of like lots of different kinds of drugs in this setup to just see how different drugs affect brain-wide activity. Because I'm not like 100% clear on like why, why they like what the rationale was for just like looking at the whole brain of a mouse while it's on ketamine. But they have data in the paper where they did it with other drugs. So I think they might just be screening like a ton of different classes of drugs to kind of get a better idea of how different types of drugs affect brain activity. Okay, so in the bottom row, panel D, you see a heat map of cortical activity 10 minutes after 50 milligrams per kilogram intraperitoneal injection of ketamine. Intraperitoneal just means they like put it in your abdomen, like in your peritoneal cavities, so below your diaphragm. So which I've done to many mice and rats. And then this is what they see. And so you can see like there's not really a lot going on, like a lot of changes going on in most of the brain, but in the retrosplenial cortex, you can see that there's this rhythmic oscillation of activity. So you can see it kind of go like up here, peak here, then go back down, then hit a trough around here, then go back up. Yeah, I mean, so I'm a neuroscientist who works with rodents as a model organism, and like this is the kind of data that like you dream of getting, because it's a really big effect, and it's very clear, and it's very clean. Okay, so they decided to take a closer look at this cortical activity, because they're imaging at a relatively low magnification, so they can't see the activity of single neurons with this setup because the resolution just isn't high, just high enough. So they're basically sacrificing resolution. Oh, sorry. I might have to switch to my headphones here. So
No
yeah, well, my headphones are right here. But we'll see if there's more noise. And then I'll put on my headphones. Okay, so basically, like at this resolution, they can see the whole surface of the brain, but they have to go in at a higher magnification or different microscope to see single cells. And they wanted, and so with this calcium sensor that I talked about, the temporal resolution is also not that great. So you can tell, okay, neurons are more active, but the temporal resolution of these measurements with these calcium sensors is due to just the nature of calcium buffering in neurons, like the signals. There's a lot of calcium buffering proteins and other, and also due to the limits of the indicator itself, like how quickly its fluorescence changes in response to changes in calcium. So for all these reasons, like calcium sensors don't have that high temporal resolution, and so neurons are firing here and not here, but you don't know exactly when they're firing. So like if you have an oscillation like this, for instance, um, it's the temporal information that you can get about the oscillation from this technique is not as high as with not as high as with other techniques. And so here on the left is again the data from the wide field calcium imaging. So on the top row, figure E, are all the different cortical regions that we just saw on the last slide that they recorded from. And the red is post-ketamine, the blue is pre-ketamine, and the difference between retrosplenial cortex and the other comparison areas of cortex is extremely obvious. So there's this really prominent oscillation in retrosplenial cortex that's not present in any of the other areas. And then here is a power spectrum of the different types of oscillatory activity. And this is still with the calcium sensor. Yeah, so this looks pretty normal for the brain. You have a lot of like low frequency activity, and then it kind of drops off at higher frequencies. And this is due to like various slow oscillations that involve a lot of the brain. But then, after ketamine here, now you can see there's this huge increase in power in the delta range, which is from 1 to 3 hertz. And delta waves are also associated with phenomena like slow wave sleep, which is essential for being able to function normally and stay healthy. Okay, so here is the quantification of the change in power. So you can see it's just a gigantic difference. Ketamine is really doing something here that the other parts of cortex aren't doing. So then here on the right is just the group mean of the time course. And this is pretty similar to the time course of the drugs, like psychoactive effects. So that's also pretty cool that it's consistent. And again, this is like amazing data. I mean, the individual traces, like I'm totally happy if I have an experiment and the variation in between the individual animals is only this much. This seems really interesting. They found this area that's like really turned on by ketamine, and but not other cortical areas. Then they decided to go to a different technique to investigate this further. So up in the upper left is just a schematic of what they're doing. So again, they have a mouse under a microscope, but now they're using two-photon laser scanning fluorescence microscopy to image the neurons. And so this approach allows, this is a different microscope, and this approach allows two-photon imaging has the resolution to enable single cell imaging like relatively deep in the brain. So in panel B, what you see are pictures of the dorsal cerebral cortex. So if you remember from the beginning, the retrosplenial cortex is located along the midline in the posterior part of the cortex. So what you're seeing here now is pieces of coronal sections of the cortex. So basically, if you sliced if you sliced like this, like along the horizontal axis of this picture, then you would get a section like this. And so here's the midline right here on the left, and then the top of this picture is the dorsal part of the cortex. And what you have here are just two different mouse lines where different types of neurons have been made genetically accessible by cell type specific expression of this recombinase called Cre. I'm not going to get into the details of this genetic manipulation technique. It's very common in molecular neuroscience. But basically, by putting Cre into neurons, then you can like make the neurons able to express DNA whose expression depends on the presence of Cre. So that's the simple way to say it. So basically, that what they can do with these mice then is they can deliver the calcium. And so these two markers, CUX2 and RBP4, CUX2 labels the layer 2-3 neurons in the cortex. So they form a layer here pretty close to the surface of the cortex, whereas RBP4 labels the layer 5 neurons. And I think they just put these pictures in there just to show, hey, like we can express our the calcium sensor that we're using selectively in these two different layers in the different mouse lines. And then they take these mice that have the calcium sensor in these two groups of mice that have the calcium sensors in the two different groups of types of cortical neurons, and they image them under ketamine. So here in H, they're looking at the change in power and in the delta range, and they find that two other dissociative drugs, PCP and MK801, also increase power. And yeah, I think there is like human data where they gave people MK801 and then asked them like published data where they gave people MK801 and asked them what it was like. But even if there isn't, like, there's definitely been people experimenting with MK801 not as part of controlled studies, and like the reports about that are that it's a dissociative similar to PCP and ketamine. So, however, memantine, which is a chemical relative of ketamine, that does not induce dissociative effects, does not induce this increase in power. So memantine is like a really good control here because it's similar chemically to ketamine, but it doesn't, the one important effect they try to study here, like memantine doesn't have it. So it's really like, a very, very almost perfect control. Okay, so what you see in I now so now in I, they're looking at activity in individual neurons. And so red means no activity, red. Blue means activity below that's below baseline, and white is activity that's above baseline. And so each of the rows is a neuron, and then the x-axis is time. And you can see that in layer 2-3, the ketamine does not really have, or the dissociatives do not really have much of an effect. But in the layer 5 neurons, there's a very clear oscillation in the delta range. And so you can see that summarized here. And then in J, they wanted to use another method, to show that that dissociatives really activate retrosplenial cortex. And so in J, what they show is data from, so again, this is the top of the brain. This is the midline right here. So they took a mouse line where in which you can turn on labeling, turn on genetic labeling of cells that are activated within a certain time period. And so that's what they did. They basically turned this system on while the mouse was on ketamine, and then later sacrificed the mouse and took the brain and saw, oh, look, there were lots of neurons active in retrosplenial cortex, like while the mouse was on ketamine. And layer 5, for those of you who aren't circuit people, layer 5 is the major output layer of cortex. So like the layer 5 neurons are sending projections like all over the brain, including to the spinal cord. Well, probably there's not that many spinal projections in retrosplenial cortex, but yeah, they're sending projections all over the brain, and the motor neurons also to the spinal cord. Okay, so now I was a little bit too early with my talking about electrophysiology versus imaging, but so here they switched to electrophysiology. So now they're sticking electrodes in the brain and recording from it. So in panel A, they just, as I mentioned, calcium imaging does not have that great of a temporal resolution. So you can tell neurons are becoming more active, but you can't tell exactly when each neuron is emitting each individual spike. With electrophysiology, that's not a problem. Electrophysiology is much higher temporal resolution than calcium imaging. So in A, they stuck some some silicon probes in the retrosplenial, in the deep retrosplenial cortex. And so in A is a raster plot. So on this is similar to the plots of the image neurons we saw in the previous figure. So basically, each row is a recorded unit or neuron. It's commonplace to call things that we think that recorded with electrophysiology that we think are neurons as units. So anyway, each of these rows is a neuron. And each of these little bars on the raster, like the little vertical bars, is one action potential, one spike. So you can see pretty clearly that when you do have the temporal resolution to really measure exactly when each individual neuron, when individual neurons and retrosplenial cortex are firing, you can see there's a really clear oscillation here. And then in the bottom row is the average firing rate of the retrosplenial neurons. So yeah, this basically confirms and extends their calcium imaging findings. I mean, this is really very prominent oscillatory activity. So then in B, they wanted to look and see, they want to look throughout the brain and see, to see if there are any other parts of the brain that display this kind of activity, and if so, how synchronized they are with the retrosplenial cortex. So in C is their recording setup. So they took these cutting edge probes called neuropixels, and so these are like some very advanced. These are very advanced. Electrode systems based on CMOS chips, and so with this technology, you can have, I think, more than 1,000 recording sites per electrode. So each of these vertical bars in panel C represents one neuropixels probe that they stuck into the brain for recording, and then the color coding just represents what area, I think, is recorded on each stretch of each neuropixels probe shank. So indeed, they just show the changes in firing rate under ketamine of all of these different areas. And then in E, they show the correlation of different regions with retrosplenial cortex. And then in F, in the plot in F, they're basically doing just a cross correlation of the activity, of all these different regions. And so in the left is before ketamine, the right is after ketamine. You can see, of course, that the cross correlation in retrosplenial cortex goes up like crazy because it's generating a ton of oscillatory activity under ketamine. And then in G, so here they're imaging three parts of the thalamus. Recording recordings from three different parts of the animal, three different parts of the thalamus. And these parts of the thalamus are all known to be important for kind of alert wakefulness and kind of non-dissociated kind of behavior. And so you can see before ketamine and then after ketamine. And so what's interesting here is that the ketamine acts, so you can see the on ketamine, the correlation of the anterior ventral thalamus and the lateral dorsal thalamus is hold on a second, I just need to check the figure legend for 1 minute. Oh yeah, I'm sorry. Okay. I think I just said something wrong. So hold on. Why can't I? Yeah. Okay. Yeah, so yeah, green here is retrosplenial recording, and then the purple is the different thalamic areas. And so you can see after ketamine, the retrosplenial cortex is like really synchronized with the anterior ventral and lateral dorsal, but not the anterior medial thalamus. And so you would never, really see this in well, or it is thought that you would not see this like in you know, under, normal conditions. And so this could be a neural correlate of the dissociated state, like that you have, some of your thalamic nuclei, that are important in wakeful behavior seem to be normal, and then seem not to be affected by the ketamine, but others become extremely synchronized with the retrosplenial cortex. So this gives us some beginning insight into the circuits that may or may not be mediating dissociation. Okay. So then, of course, they wanted to in order to do kind of further characterization of this phenomenon, they really needed a behavioral correlative. They needed better readouts of dissociation. And so they turned to behavior because, I don't see any other really way to know if a mouse is dissociated. You can't ask them, so you got to do a behavioral paradigm. And so they actually had to develop a behavioral paradigm in which there were really dissociative specific differences in behavior. So this is also really cool that they kind of extended the hot plate test in a way that enabled them to really distinguish different effects of different classes of drugs. So I thought that was really cool. So the hot plate test is which is panel A of the first panel. So this is a pretty common test in pain research, and it's also used in other areas of neuroscience. So the basic idea is, you're trying to test the mouse's pain response. So you expose it to a hot stimulus and then see how well its reflex works. Because 55 is hot enough that they don't like it. Okay, so then what they did was they kind of broke it down into two components. So one is just the reflexive, take your paw away from the hot plate. So this is the reflex part of it. And then there's another part that's the affective or emotional response. Like, what do they do after? Well, normally they think they might have been burned, so they grasp their paw and they lick it. So then of course they did a whole dose response of ketamine and showed that it maybe a little bit impairs the reflex, but not significantly. And so this really shows that like ketamine's like not a great analgesic, and that its ability to block painful stimuli comes mostly or entirely from anesthesia. Okay, so then they also looked at the licks per minute, and here there was like a really big drop at the higher doses of ketamine, which are also, I think these would also be the dissociative doses, and the lower doses probably are non-dissociative doses. Again, we can't know for sure because we can't ask mice. But I mean, this is kind of consistent with, how people have modeled, pharmacokinetics between mice and humans. So then in C, this is a recording from the retrosplenial cortex, and you can see at the low dose there's no oscillation. At the higher doses there is, and this is just a summary of the power, the spectral power. And then in D they just show that the effect on the power of the oscillation is also dose dependent, and it seems to reach a ceiling at doses of ketamine where the mouse is probably in a K-hole. Okay, so then of course, this isn't enough. This isn't enough though, because they really, with this assay, also want to show, like, they want to have an assay that can measure effects. Sorry, that can measure effects that are specific to dissociatives and not shared by other drug classes. So on the right is a comparison they did between drugs. And this is why I got the idea that they must be just screening lots of different types of psychoactive drugs in this whole brain, with this whole brain imaging setup. So so basically they used, PCP, another dissociative, buprenorphine, that is an opiate analgesic, lidocaine, that is non-psychoactive relative of cocaine, but it's still a painkiller. LSD and diazepam, and then they have more data from other kinds of drugs in the supplementary if anybody's interested. Yeah, so there were a lot of differences between the drugs, but you can see the effective PCP also really got rid of the effective response. So sorry, here in the E is just the reflex. So different drugs appear, like as you would expect, the painkillers and the sedative impaired the reflex more than the other drugs, which actually didn't impair it at all. But the kind of grasping and licking the injured paw, the effective response, PCP really suppressed that just like ketamine. So and also these other drug classes failed to suppress the effective response. And I mean, this is a dose of a benzo where the animal, like, it's interesting because like the animal will have a, this is a dose of the like, for instance, diazepam, where the animal definitely has a pretty big impairment in the reflex. So like it's unlikely that there isn't enough drug on board to impair this process if it was important. Okay, and then here in F, so they put the mouse in an open field, which is just an open arena where they can run around, and they monitor they monitor their activity. So on the right panel here are examples of trajectories of animals in this arena. And so the gray trajectory is I think before the injection or on another day from the injection, and the black trajectory is after the injection. And so you can see the animals, when they get dissociatives, they don't explore the environment as much. Actually, I'm not sure I remembered the legend to that correctly. But yes, and then you can see also, so you basically see that that here there's really no consistent effect on activity. So PCP is a lot more stimulating than ketamine, and so therefore it doesn't really cause the same decrease in just general locomotor activity than ketamine does. So yeah, I mean, it seems like this assay, they found like a behavior, this paw grabbing and licking the injured paw, that seems to really be selectively impaired by dissociatives. And this behavior is impaired even when in dissociatives that have different, that have opposite effects or on other behaviors. So this is pretty cool. Now we have a probably quite useful new behavioral paradigm for studying lots of things, actually. Basically now they can use this behavior to study the neural mechanisms of dissociation, of drug-induced dissociation. Well, the first thing they did, of course, was they wanted to see is if we stimulate these neurons in retrosplenial cortex, can we induce a dissociative state? And of course, to do this experiment, you need a way to measure dissociation in mice, which is what I just explained to you. So basically in this experiment, they use optogenetics, which is a technology that allows control of neurons firing with light. And so using these mice that I told you about earlier, they could target their, their light activated proteins specifically to the layer 5 cortical neurons in retrosplenial cortex. And then they just did, so with blue light they were stimulating the neurons, and so they stimulated them in these 250 millisecond bursts. And then with yellow light they were inhibiting the neurons, so they're basically just trying to like force the brain in, like force the layer 5 neurons of deep retrosplenial cortex into this oscillation as much as they possibly could. And yeah, so then in here you have the in the middle you have the retrosplenial cortex data, so you can see when and then in the gray is just a control, so where they put where they virally transduced just a fluorescent protein into the neurons, but without another protein that enables you to control the neurons with light. Yeah, so so in retrosplenial cortex, when the stimulation is rhythmic, then the licks go down, but again, the flicks don't. However, when the stimulation is random, so when the kind of the times of the lights being turned on and off is shuffled, so that you don't get this rhythmic oscillation in the stimulation pattern, then you also don't get a decrease in the effective licking. On the other hand, when they did this in somatic, when they looked at the activity in somatic sensory cortex, it doesn't change at all, regardless of the conditions. So with this experiment, they really show that you can associate, you can elicit the same behaviors that are elicited by dissociatives when you stimulate this part of the cortex. So this is, which is pretty cool. I would not go so far to say as you can actually elicit dissociation because with just from this data. So, and of course, I already mentioned circuitry. So they were also, they are also interested in what inputs might drive this activity, like where is what other part of the brain is the information coming from that sets up this oscillation in the retrosplenial cortex. So here in A, they have a diagram. So the red is the retrosplenial cortex, green is dorsal thalamus, and blue is subiculum. And the dorsal thalamus and subiculum are deep brain areas. So they're actually like millimeters below the surface of the brain. So basically there's kind of at least two ways that this oscillatory activity could be induced, just locally, like within the retrosplenial cortex. One is through, and that is through different ion channels on the retrosplenial neurons. So these so HCN1 is a neuron is an ion channel that's been shown, that's where there's quite a lot of evidence that it's important for setting up oscillatory activity in different parts of the brain. The NMDA receptor, in addition to being the pharmacological target of ketamine and PCP, is also involved in generation of oscillatory activity. So yeah, they kind of wanted to know, okay, are other parts of the brain providing this information, or maybe it's just through internal, dynamics internal to the area, internal to the retrosplenial cortex, based on the intrinsic membrane properties of these neurons. So here in B, they're showing the results of anatomical tracing experiments. So they made an injection of a virus encoding a fluorescent protein into these different parts of the brain, and then B, they're just showing, okay, here's where our injections went. So here's the retrosplenial. Hold on a second. Yeah, so here's the retrosplenial. Here's cingulate cortex, which is just in front of retrosplenial cortex. So they just wanted to show that like the injection didn't bleed into the cingulate cortex. Here's the subiculum, and here's dorsal thalamus. And so the first thing they did to try to answer, to answer this question here in the upper left of panel A is to record to record bulk calcium signals. So again, they put the same calcium sensor into the retrosplenial cortex. So here you can see that, which is depicted here, and then and then they then used optogenetics to inhibit either the dorsal thalamus or subiculum. So basically, they could turn it off for however long they wanted by inhibiting the neurons. And so that's depicted in this front section in C. And so what they found was that when you take out the subiculum, nothing happens. Ketamine doesn't do much, but this is the power spectrum. So they're measuring the power of the oscillation here. However, when you take out the thalamus, then it has a pretty big effect. So the ketamine really does stops. Ketamine stops eliciting this oscillatory activity as much when the thalamic inputs are inhibited. So then in D, in this right part of the slide, they're addressing this question that's in the right panel of A. So here they're using, so basically want to look at the contribution of these two receptors, the NMDA receptor, GRIN1 is the gene for the NMDA receptor, and the hyperpolarization activated cation channel, which is HCM1 is the gene for that. So what they did was they used this same genetic system I described to you earlier to selectively knock out these genes only in the retrosplenial cortex. So this will really enable them to answer the question of whether those receptors or ion channels are important specifically in this part of the cortex to generate the oscillation. So in E is the setup. So they don't show the local knockout, but basically they have, it's just a viral injection into a transgenic mouse to knock out inject this Cre recombinase into specific brain, into the retrosplenial cortex of a transgenic mouse that's been engineered so that, your gene of interest can be cut out by Cre. And so in D, what you see is just the expression of these ion channels in retrosplenial cortex. So this is the top of the brain, is the top of the picture again. So you can see that there's lots of NMDA expression in retrosplenial cortex, and then there's also HC1 expression pretty clearly in cells. And so then what they did was then they so they basically put an optic fiber into the retrosplenial cortex of these mice where they locally knocked out the channels, and then they recorded the bulk calcium signal to see if they could still see an oscillation. So and of course they also looked at the behavior. So control animals, before ketamine nothing, after ketamine oscillation, and then in neither of these two knockouts, in both knockouts, the oscillation was impaired. However, in the HC1N1 knockouts, it was impaired more. So there's still a significant difference between pre-ketamine and post-ketamine in the NMDA receptor knockouts, but then in the HCM1 knockouts there is no difference. But in any case, it seems like probably the HCN1 channel is the biggest contributor to this oscillation. Although, like, from the data, it does look to me like they probably are working together, and they're really probably both required. And so in the behavior, this is actually a little bit of a mismatch here, because in the behavior, you can see, again, the reflex is fine, and the licks are actually rescued when you knock. So basically, when you make the retrosplenial cortex unable to oscillate by knocking out this channel, then the animal displays normal licking behavior after getting its paw burned up. After the painful stimulus on the hot plate. And so this is okay, why is their control data 0?
Peaks do go down, right, with ketamine?
Yeah, well, yeah, I mean, sorry, I'm not sure. Yeah, I thought this figure represented one thing, but I think it's representing something different, actually. Oh, wow, I'm taking a ton of time to go over this. I need to speed it up.
Hey, no worries. Like, you're going through a lot of things as well. Like, this is a lot of information. I think it's a good pace, actually.
Okay, well, yeah, and I'm a little nervous that, like, there have been so few interruptions, because, like, that makes me worried that people aren't following it.
No, I was saving them for the end, because you were, I think you were going through it very nicely.
Okay.
I have some questions about this one if you want them now.
Yeah. You can go ahead. Oh wait, no, yeah, I'm sorry, I got myself confused. These mice have all had ketamine, so yeah, of course. Yeah, sorry about that. Yeah, so basically, like, in the HCM1 mouse, the licking is rescued. And it would have been really nice in this data to have another group of animals where they don't give them ketamine. So then you can see what the baseline level of licking is. Because, like, I really want to know if this is a partial rescue or a full rescue of the licking. Because based on their electrophysiological data, what I would expect is that in order to really bring the licking back up to baseline, you would need also to knock out NR1 in the retrosplenial cortex.
Maybe if you go back to the previous, so this one says 4 licks per minute. Maybe if you go back to the previous slide, did they have licks?
Yeah, they did, but it's a different experiment, so, like, you can't really directly compare them.
But I mean, here it seems like it's more like in the control condition. Well, or no, this is a different thing. Yeah. But it looks like it's more in general, right? In more than 4. So maybe, as you said, it's kind of a partial effect.
Yeah, I mean, it's you can't really say unless you have the other experimental group where they didn't get ketamine. Yeah, because the control mice is just basically, the control group basically just says here, okay, ketamine is doing what we wanted to on the effective response.
But can I ask you something about this one?
Mhm.
So here, the part C, right? It seems to, if I understood correctly, it seems to say that basically the thalamic input is relevant. Let's say it's important for this effect of ketamine on the oscillations, right? But also, I think a few slides back, we said they were saying, so it was quite clear that this rhythm is in the layer 5, right?
Oh, yeah. Okay, yeah. So that's a good question. So basically, yes, they show here that these neurons are in layer 5. And then, but these, but yeah, I mean, something has to be, driving the activity in those neurons. Or well, probably.
Yeah, that's what I was want. Like the idea here would be that the thalamic input towards retrosplenial cortex is kind of, well, yeah, it's
Yeah.
involved. Let's say it's hard to. I think that's also why in the title they don't really have a verb, right?
Mhm.
It,
Yeah.
anyway.
Yeah. So yeah, this basically shows that it is causally involved in the
Yeah.
oscillation, so that it may help. It's evidence that that the thalamic input actually like plays an important role in generating the oscillation.
And do you know what when they do these experiments, like what kind of environment are the mice in? Like,
That's a great question. It's in the methods. Hold on. I hope it's in the methods here. Okay, so they are. Okay. Um. Hmm.
I mean, otherwise it doesn't matter. I was just wondering, since the thalamic input seems to be important, so it doesn't seem to be just a local, like only a local dynamics phenomenon, right? It seems like
Yeah. Yeah. Yes, it does not seem to be a local dynamic. Okay, here we go. Okay, so basically what they did for the hot plate test was they had like a circular wall. So they basically have the hot plate, and then they have like a circular wall around the hot plate, and then they're videotaping from two different angles, and they just put the mouse on the hot plate, and then see what it does. And then if the mouse doesn't do any, if the mouse does not escape within 90 seconds, they remove the mouse. So that's how they did it.
all right. Thank you, Rudy.
Um, yeah. So all of this is really cool and interesting, but does it apply to people? Well, fortunately, in the medical center at Stanford, they had an epileptic patient with dissociative auras, and the epilepsy was so severe that they had to implant electrodes in this patient's brain in order to figure out, if they need to take a part of the brain out, and if so, which, basically to try to find this focus of the seizures and see if they could take it out. So in A, they're just showing some example depth electrode recordings from this patient. So I don't know if you can see this, but there's like these little columns of dots in these schematics of the patient's brain, and those represent different probes, and each dot is a different recording site on the probe. So and then basically the purple recordings are deep posterior medial cortex, and the black are other parts of the brain. Okay, so basically here in A, they're showing recordings from a seizure in this epileptic patient that has dissociative auras. So you can see pretty clearly that in the deep posterior medial areas of cortex, which are thought to be homologous to the rodent retrosplenial cortex, there's a very clear delta oscillation during this aura. And you don't really see anything similar in any of the other parts of the brain they were recording. In B, there are some descriptions from the patient about these auras. So I was listening to two parts of my brain speak to each other. In a way, the third part of my brain, which I considered to be me, was able to listen. So that's like, that sounds pretty typical for. Well, I mean, it sounds like something that's, things like this are often reported by people who have dissociative experiences. Yeah, and then what would it feel like if someone else were to come into your head? What I considered me shrank to this other part of me, where the other parts of my brain that were talking, I stopped considering them me. So yeah, he's dissociating from these other parts of his brain or himself. And then in this last one is an example of more like physical dissociation. Like the K-hole. So like, where in this 3D space am I? I took a blanket, I threw it over my body just to see, because I know that when I don't feel it, I don't consider it me. And immediately my legs were no longer a part of me. And then in C, they're just showing when the onset of the seizures was scored. So basically they had neurologists score, okay, this is when the seizures started. So again, there's this really prominent oscillation in the deep posterior medial cortex in the patient, and in none of the other or recorded areas. And this is definitely happening like before the seizure. So because they had all these electrodes implanted in this patient, they could also stimulate at the recording sites. And I will get to that in a minute. So in D, they're showing these are in D and E are recording data from recording the activity of the neurons on these different probes they put into the patient's brain to localize the seizure focus. So again, these are power spectra, just showing how much power in the activities at different oscillations. So during the aura, there was a big spike in delta power in this in deep posterior medial cortex in the patient, but not in superficial posterior medial cortex. So this is similar also, like into the mouse data, where like they see these effects in the deeper parts, but in the mice they also see them in the superficial parts too. So this is also a bit different. Okay, then they also looked at. Sorry, this is now the left side, and the left side also goes up. During the aura, and you really don't see. You really don't see that in the non-seizure conditions. And so this patient actually, they localize the seizure to the epileptic focus to the right side of the brain. So which is important for panel F. And then in panel E, they're just looking at the power during the aura. So it's really much higher. So these first two columns are the deep posterior medial cortex on the left and right side, and the power here is just so much higher than any of the other areas that they recorded from, and also the difference between seizure and non-seizure activity. Well, I should say aura and non-seizure activity. So as I said, they stimulated at these contacts. So here are 3 comments by the patients. The right one is where the right side is where the seizure is. The seizure focus is. So 1, felt similar to the beginning of a seizure. Felt like an aura. It's like I'm about to have a seizure. Not super pleasant. So they also decided to stimulate the other side, where there was no seizure focus. And so now he said, okay, so now, they hopefully, their goal with this was to enable him to elicit dissociative experiences without the negative, oh no, there's a seizure coming feeling with them. And and they were able to actually, based on what they report, they were able to do this successfully. So when they stimulated the left deep posterior medial cortex, they had some, the patient described things that were more similar to, let's say, drug experiences instead of seizures. So, this feeling of being disconnected from something, that was a little pleasant. It's like being weightless in your own mind as a personality. And maybe the same way a pilot can lose control of a plane, like they can be forced out of the cockpit or to not control it, but still see what's happening to the whole plane. That's kind of what just happened. I got pulled out of the pilot's chair, but I can still see all the gauges. You can still see the information flowing. You can't control it, but you can see it. I like this last one as a description of the dissociated state. It's a pretty good analogy. Okay, and then finally they quantified. So they stimulated not only here, but on other recording sites. And so now they're just on the y-axis plotting the percentage stimulations that could cause an aura. And so basically sham stimulation is where they have an electrode in there but don't stimulate. All other cortical areas, only one stimulation could generate an aura, whereas in the deep posterior medial cortex, 11 out of 13 stimulations generated an aura. So that is some really cool human data, and it's summed up, well, all the data is summed up here. So based on this tour de force of work, this is how you can induce dissociative states experimentally. So with the mouse, again, to really force the retrospinal cortex into this oscillation, you can inhibit neurons with yellow light and then activate them with blue light alternately. So to basically force all the neurons only to be active like on the period of the oscillation. And so you get some behavioral effects that are specific to dissociative drugs and mimic the effects of ketamine. And then in the human, so yeah, they can't, of course, do the optogenetics because it's quite invasive. So and also unproven for therapy. So here they just electrically stimulated in the deep posterior medial. Oh, whoops! What did I do? In the deep posterior medial cortex, and then they found that the patient reported a dissociative experience that was very similar to their auras. So there's a really excellent cross translation between mice and human in this paper. This is kind of a dream for anybody who does translational neuroscience. Yeah, so I don't know. I think this was I already said it was a really excellent piece of work. Yeah, I think it could be improved. I think the question I'm left asking at the end is, like, what aspect, is their behavioral test measuring like a clinically relevant component of the dissociative state? So, yeah, I mean, I really want to know more. I really want more evidence about how well like their mouse model translates to specific components of the dissociative state. But still, it's an amazing piece of work. All right, and that's it. Anybody else have questions?
Yes. So
Go for it.
no, I mean, just wanted to first again thank you, Rudy, for giving this
You're welcome.
presentation. I think it was super interesting, and as you said, it's really nice work in many ways. Yeah, I guess the other questions that I had was the ones that I was pointing at, like the role of the thalamus in this whole picture, let's say, and also the roles of those I think the same things that you were pointing out, like the fact that there would seem to be like multiple channels that kind of had, when they did the knockout stuff, that kind of, to some extent kind of different channels that cooperated to or were needed to create this effect. So those are the things that I'm left kind of wondering about. But as you said, it's really like beautiful, beautiful data and beautiful combination of methods.
Yeah, I mean, I'm kind of wondering about like their interpretation of the channel knockout experiments, because maybe they just wrote it this way just to make an easier, a more digestible story. Like, okay, we found one gene, this is it. But I mean that's really not what their actual data suggest. Okay, maybe HCN channel is necessary for the oscillation. Like you can't have it without the oscillation. Like you can't have the oscillation without it. Like I think they showed that pretty well. But I mean, I definitely think like probably the NMDA receptors also. You're not gonna, yeah. I think if this oscillation is really important for the actual state of being dissociated, then like I think you wouldn't be able to you wouldn't be able to like dissociate a mouse if it didn't have you wouldn't be able to put the mouse into the cognitive state or behavioral state of dissociation without also having the NMDA receptors in the retrospinal cortex.
Yeah, I just wanted to say, I think this, what a fabulous presentation. Thank you so much for all of the work you put, or I don't know how much work you put into this, but it felt like a lot, and you managed to just cover so much within 1 hour. And explained everything so clearly. So yeah, thank you for that. That was really great.
Thank you.
I was wondering about the last slide. There's so there's the retrospin. Also, is there any sort of association with the retrosplenial cortex? Like, what's the known function of it?
Well, that is it does have functions. Okay, so I have no idea like what cortical region in the human brain, what that part, that the deep posterior medial cortex, I don't know what that does. I think it's part of parietal cortex, but maybe it's part of the temporal lobe. I don't know. It's pretty deep. So like retrosplenial cortex is involved in associative learning in mice. So it is important for fear memory. It can be important for fear memory. I think for more remote fear memory, for long-term fear memory, retrospinal cortex is necessary. Yeah, I think what is, I'm no expert on retrospinal cortex, but I think based on what I've picked up, over the years, it's one of the places in the cortex where long-term memories go for storage.
Gotcha. It sounds similar to like they're doing like fear extinction or something, training with ketamine now with people. Is what I've heard. I don't know if any of you guys have heard of. Yeah. I guess phobia extinction or something.
Yeah, in Amsterdam.
I'm not sure. I just recently heard this, but
Kint's lab at the UvA has done really great work on using beta blockers to cure people's phobias.
Yeah, propranolol.
Any more questions? Any final questions?
Yeah, I have a question. Thank you for the presentation, and I can certainly see why it's kind of exciting data, and I can agree that it seems like something that will have an impact. And I wanted to ask you, Rudolf, if you could speculate a bit about where this might lead. Like at the moment it's kind of abstract and it's in an animal study and this kind of thing, but could this lead to some sort of practical application in some sort of therapy for some sort of disorder, or what do you see for the future of this?
That's a good question. So, well, I think hopefully this provides a first step toward being able to identify dissociated states without behavior, or especially without a patient having to report to you, because like if they're dissociated, maybe they might not be able to make a report to you, or not be able to make a report that's intelligible. And let's see. Okay, aside from understanding the brain better, like what other practical things could result from this? Okay, I'm just gonna wait for the doorbell to stop ringing. Maybe for screening drugs, if like the recording experiments could be like, if the throughput could be increased, then like I could see it maybe being used to screen drugs for dissociative effects. Like for drug companies, if they're developing a psychiatric drug, to make sure that it doesn't have the unwanted side effect of dissociating the patient. Let's see. Let's see, other practical applications. So they know, I mean, okay, so let's say, I guess also they like, there's pretty good evidence now that this, the deep posterior medial cortex in humans is involved in generating dissociative states. So this is actually a really interesting question for myself. Like, I want to ask my psychiatrist friends, but now you have a way. Now you have a part of the brain where, like, let's say you have a patient with dissociative symptoms that's causing problems in their life. Now you have a part of the brain that you can target for potential therapies, like maybe deep brain stimulation or something like that. Let's see, and then.
I mean, if I may, just one. So you've seen that ketamine, is being used in like treatments in like clinical, in some clinical applications, like for depression. So one thing that this could be relevant for is to like try to see whether this kind of some this if this dissociative experience is as indexed by the the rhythm in the retrosplenial cortex, if it's something that is relevant for those like therapeutic outcomes, or if it's something that is not that doesn't correlate that much. So then you if you see that it seems to be like relevant, then you could also like try to see if it's a more general principle, and also if inducing, I don't know if maybe not, I don't know if how much of the dissociative experience is relevant for like therapy, but if it was, then you could try to induce that in some other way and see if that also has like therapeutic benefits. Or if it's not, then you could try to avoid it, if it's like not a useful component, let's say, of the therapeutic effects. So I think kind of like Rudy was saying, this kind of also could help disentangle let's say the clinical benefits of ketamine.
Yeah, absolutely. Yeah. So yeah, I mean, if this is, and they did give, yeah. So I guess of course, yeah, one of the most obvious questions is that, as Marco said, is like, is this oscillation required for the therapeutic effects of ketamine? And I don't know. I'm I don't know I don't have enough knowledge of like pharmacokinetic translations between species to really like have a good idea about that. I mean, based on what I don't know, I think, I mean, I wouldn't be surprised if like it needs to be like, a K-hole level dose to get this rhythm. Because I think in one of the earlier figures, they had some effect on some behavior with, nope, no, actually, no, they didn't really have I just remembered it wrong. They really didn't have a graded effect on behavior at all. The lower doses. So yeah, but I mean, I think for also I think for treatment of psychiatric disorders that involve dissociation, like you could use this as maybe as a clinical diagnostic. Like if patient presents, you're not sure what's wrong with them, you can like throw them in. Throw them in the fMRI, if you can even use that for diagnostics, and like see if the retrosplenial cortex is lighting up. So like there's a potential that like, it could be used for diagnostics. I'm not sure actually how widespread that use would be, but it's something.
It's interesting because you said that earlier in your talk that within a clinical setting, the dissociative state is kind of defined very broadly and can be many different things. So I wonder if this will bring a finer grain to that.
That's just in general, like not just the clinical setting, just in general. It's defined broadly because it can. I mean, it can. It can take on many forms, but like the fundamental aspect is being detached from, normal reality in some way.
Yeah, makes sense.
Yeah, and so, yeah, I guess one next step is to try to experimentally induce dissociated states in volunteers non-invasively. Because right now the human data is only one patient, and it's like really exciting data, but I'm sure they're trying to find more patients, but I think the neurological patients really for this can only serve as proof of principle in people, and then but then if I'm not sure ultrasound stimulation is at the point yet where it can reliably stimulate something somewhere that deep in the brain. But if you could reliably induce dissociation in people by non-invasively stimulating the deep posterior medial cortex, then like that would open up a lot of possibilities, for interesting neuroscience experiments, and also really confirm beyond a shadow of the doubt that this is a core component of the dissociation circuitry. And then dissociative symptoms are a part of psychiatric disorders like PTSD. So like maybe understanding this circuitry can help us to manage the symptoms of those disorders better.
All right.
For instance, like maybe this oscillation will end up having good predictive validity for therapeutic outcomes, for therapies for dissociation. That would be pretty cool. Okay, I think I'm done with all of my commentary now.
No, it was really interesting again. I guess for the more neuroscience inclined people, but that's how it is.
Mm-hmm.
So I think I would wrap it up and stop the recording now.